If I double-clicked on that diagram and said, what is needed underneath the tool in order to support this? Well, it's a complex database. This is, again, a sort of high-level description. The real database has maybe 10 times as many entities in it. But this diagram, the seed, model seed databases, shows the relationship on the right to a system we built called the seed. And if you are sitting there at your machine, you can Google PubSeed, P-U-B-S-E-E-D, and you'll take you to a website that is the web front end on the public version of the seed, where that database essentially has genomic data all the way through functional gene function data, and then the corresponding database called the model seed, which is actually a database of entities needed to support modeling. And so these two databases are integrated through a set of entities that span them, span protein sequences and IDs, gene-protein relationships, and so forth. And so these two databases are what we need to do to implement that pipeline I mentioned before. Next slide. Jeffrey, this has a couple of pieces, so you should click twice on it, or whoever's moving the thing. So just to give you a scale of these databases, the seed database, the genomics database, has, actually it's about 5,000 genomes now, and an additional 30,000 non-public genomes. One thing we've discovered as we made these systems available, and we made them available in two ways. One is for people to access the public data, and the other way is for them to use them privately to annotate their genomes before they're published. And what we discovered was that right now there's roughly 10 times as many private genomes in circulation in some sense than the public genomes. And this represents an increasing sense that it's increasingly difficult actually to get genomes submitted into the public archives. Many groups don't even bother to do that anymore. They do the analysis and their results never get public. So one of the challenges we have is how can we make it easier for people to publish data? That's an important sort of side effect of this. Same thing is true in the models. Of course, we build models for all the public genomes, but we have over 10,000 models that have been built by individual users that are not yet public. And you can see the number of compounds and reactions and so forth that are in here, including a library of almost 600 different media conditions for virtually growing your organism. Next slide, please. I won't go through, this is another pipeline kind of slide. I don't want to go through it in the interest of time. These slides are available. I think you can grab them. But this just indicates that at the beginning of this, at the top of this diagram, we have an automated annotation server that you upload your genome, and then we process through a series of steps, annotating that genome, building a metabolic reconstruction, building an initial model, running some steps to correlate that model with known experimental data, additional data for consistency analysis. Then we do a series of steps called gap filling, which essentially makes a series of conjectures to add steps to the model that might be missing. In other words, these models of an organism, in order for them to be minimally complete, they have to be able to model the organism's growth. But based on what we understand of a given genome, if we can't do that, we have to add additional things to the genome to enable modeling the growth. And that each of those things that we add represents a conjecture and it becomes a testable hypothesis. Then we run a series of standardized analyses, like predicting what happens if you deleted each gene, a computational gene essentiality analysis. Ultimately, if you have the right kind of data, we can essentially automatically produce models that are approximately 90% accurate in predicting, say, the impact of deleting genes or growing on uncertain conditions. Next slide. Now, the tools I've talked about so far, the seed, model seed, RAST, I haven't talked about MenGRAS, but is part of a collection of tools, including tools being built out at Berkeley, Microbes Online, Metamicrobes Online, and the RegFam set of tools that together represent the current, in our view, sort of a very comprehensive set of tools for doing biological analysis of individual microbial genomes, collections of or samples that come from the environment that are sequenced without trying to separate the organisms. Those are metagenomes. And together, these tools have about 20,000 worldwide users. And for example, RAST, which is the genome annotation engine, this morning just got its 510th citation, and it's been up and running only for about 3 years. So it gives you a sense of the scale of these systems. Now, the challenge that we face is how do we integrate all of these together into a single environment that's more powerful than the individual collection. Next slide, please. It's titled Integrated View. I can't tell what the slide number is, but Integrated View of Modeling Simulation. So if you think about what I've just been saying, I talked about integrated biological databases, and of course, things that go along with those databases are bioinformatics tools that might be multiple sequence aligners, they might do comparative analysis, phylogenetics, etc. And these tools in our environment are interfaced with the database. So when new data comes in, we can do analysis, we produce additional data products, which then feed back into the databases. And so there's a sort of a loop that happens between the data tools back into the databases. And then we can use that system to design experiments, the blue part of this path, where we'll do experimental design that typically would include some kind of high throughput activity. We would collect the data, maybe visualize it, analyze it, and then take the output of that and integrate it back into the database. And so this has been sort of what's been possible for the last 5 years or so. But ultimately what we want to be able to do is add, click the next slide please, is add this upper level, this upper circle, that says in addition to planning experiments, we want to use this system to design modeling and simulation activities. And these modeling and simulation activities might be a parallel to the experiments that one would do. So for example, if I sequenced a new genome and I want to understand what that organism is capable of doing, I build a metabolic model, and I now run that model on a whole variety of conditions to see whether I would predict that it could grow in various compounds. And then to validate that, I would say, okay, let's now do an experiment where we're going to try to grow this organism in these different compounds. And then, as you can see on the right-hand side of this, we have to reconcile the results of the computation with the experimental results. And it's the combination of these two cycles together is going to accelerate our understanding. Next slide. So the system, this is systems biology knowledge base slide. The system that we're building now, that's the integration of all of these tools, is called the Knowledgebase, or KBase for short, KBASE. And the goal of the KBase project is to enable predictive systems biology. That is, to enable many thousands of users to build models, to run these models, to design experiments, integrate the results of experiments and modeling, and then facilitate this kind of acceleration of biology. And the 3 targets for this project are microbes, and because this project is funded by DOE, we're mostly interested in things that are not biomedical. It might be microbes that live in the soil, that participate in carbon cycle. They might be microbes that are used for bioengineering, biofuels, etc. We're interested in communities, that is not just individual single species of microbes, but the complex communities that really live in the real world that do things, again in the soil or ocean or wetlands and so forth. And we're also interested in plants, particularly from a bioenergy perspective. And of course, plants being eukaryotic organisms, they're much more complex from a genome standpoint. And the interesting collection of all having these 3 targets is that the technology is most well developed on microbes, and we're moving that towards communities and moving it towards plants. So over the 5 to 10 years of the initial stage of this project, we'll be seeing a lot more capability in dealing with model plants in the future.